Welcome to Analytical Chemistry Vision, a platform dedicated to exploring the theoretical and experimental aspects of chemistry, blending classroom learning with laboratory research to enhance understanding and inspire innovation in chemical science. Let us begin a very interesting and highly useful topic for UG, PG, and research students. The steps in chemical analysis, first part 1. Sampling. In this lecture, we will learn how environmental, food, and clinical samples are systematically processed to determine their chemical composition. In this lecture, we will learn following topics in Steps in Chemicals Analysis, Sampling, Part 1. First one is Importance of Chemical Analysis. Second one is Criteria for Representative Sample. Third one is Steps in Chemical Analysis. Fourth one is Types of Sample Collection, Bulk, Gross, and Incremental. Fifth one is Sampling Statistics. Sixth one is Methods of Sample Reduction. Seventh one is Techniques for Air, Water and Soil Sampling. What is need for chemical analysis? Why we do the analysis of soil, water, milk, vegetables, clinical samples? The reason is determined to chemical substances such as copper, nitrogen, zinc, nickel, iron, manganese, lead, cadmium, arsenic, sodium, potassium, etc. Anions such sulfate, nitrate, phosphate, chloride are analyzed in these type of samples. In addition, vegetables and medicinal plants are analyzed for determination of vitamins, minerals, flavonoids, phenolics, fats, proteins, etc. And blood and urine samples are analyzed for measurement of proteins, cholesterol, creatinine, uric acid, DNA and RNA, etc. Here, again questions arises, can we directly take samples such food, soil, fish, eggs, milk, vegetables and clinical samples in flask and proceed for chemical analysis in analytical instruments? So, answer is no. We have two following steps for analysis of chemical composition in environmental, foods and clinical analysis. First step is sampling, collect a representative sample for analysis. Second step is sample pre-treatment, remove unwanted materials and make the sample suitable for analysis. Third step is measurement, analyze the prepared sample using appropriate tools, volumetric, gravimetric, atomic absorption spectrometry, UV visible spectrophotometry, high-performance liquid chromatography, or electrochemical sensors to obtain accurate quantitative results. Let us see first step of chemical analysis, sampling. Sampling in chemical analysis is the process of collecting a small, representative portion of a bulk material, solid, liquid, or gas, to truly determine its chemical composition. Types of samples. Environmental samples, water, air, soil, clinical samples, urine, blood, tissue, food samples, milk, grains, vegetables. Here, we have to know what is representative sample. A representative sample is a subset of a population that accurately reflects its overall characteristics. It ensures reliable, unbiased analysis. Examples: if we want to determine heavy metals in milk samples of Chhattisgarh, India, then we collect milk samples from all the districts as well as include the bulks in the districts. Next, what are the criteria for representative sample? First one is homogeneity. The sample must accurately reflect the overall composition of the bulk material. Second one is adequate sample size. It should be sufficiently large to represent all components, including major, minor, and trace constituents. Third one is proper sampling technique. Incremental samples must be taken from different locations or times to avoid bias. Further, fourth one is improper mixing. Incremental samples must be mixed thoroughly to form a uniform gross sample. Fifth one is correct preservation. Samples should be stored in appropriate containers under suitable temperatures to prevent degradation or chemical changes. Sixth one is documented procedure. Sampling should follow a defined, standardized, and validated method to ensure reliability and reproducibility. And then, seventh one is randomness. Sampling points should be selected such that each portion of the bulk has an equal chance of being included. Eighth one is avoidance of contamination. Containers, tools, and environment must not introduce foreign materials. Ninth one is avoidance of losses. No loss of volatile or moisture-sensitive components during sampling. Another one is type of sample collection. There are three types. First one is increment sample, second one is gross sample, and third one is bulk sample. First, we will see increment sample or primary sample is a small portion of material collected from a single point at a single time. Examples: a 50 gram soil taken from one spot in a field. A 100 milliliters water sample taken from one depth of a lake. A 10 milliliters milk sample taken from one corner of a storage tank. Second way to collect the sample is gross sample or composite sample, which is a combined sample made by mixing several increment samples collected from different points, depths, or times. Examples: soil from 10 different points in a field mixed together to represent the whole field. Water collected from surface, middle, and bottom of a river and mixed to form one composite sample. Multiple milk increments taken from different positions in a tanker mixed to make a representative sample. Third type is bulk sample or laboratory sample, is a small, reduced portion of the gross sample that is taken to the laboratory for actual chemical analysis. Examples: from a 1 kilogram gross soil sample, 50 grams is obtained by coning and quartering for laboratory testing. From a 2-liter composite water sample, 100 milliliters is kept for metal analysis. From a 500 grams mixed grain sample, 20 grams is used for nutrient or pesticide testing. Next we will see how the statistics involved in sampling. There are sampling methods. First one is probability sampling. This means every unit has a known, non-zero chance of being selected. First one is simple random sampling. Every member of the population has an equal chance of being chosen. Example: randomly selecting 10 water samples from 100 collected bottles for heavy metal analysis. Second one is systematic sampling, selecting samples at regular intervals. Example: collecting air samples every 30 minutes in a lab to monitor VOCs. Next, third one is stratified sampling. Population is divided into strata, groups, and samples are taken from each group. Example: dividing soil samples by depth, 0 to 10 centimeters, 10 to 20 centimeters, 20 to 30 cm, and sampling from each layer. Fourth one is cluster sampling. Population is divided into clusters, and whole clusters are sampled. Example: selecting 3 villages, clusters, out of 10 and testing all their drinking water sources for fluoride. Further, fifth one is multi-stage sampling. Sampling occurs in multiple steps combining methods. Example: selecting districts, selecting villages, selecting wells for water testing. Sixth one is composite sampling, combining multiple small samples into one mixed sample to get an average value. Example: mixing 5 soil subsamples from different points of a field to analyze nutrient content. Second type of sampling in statistics is non-probability sampling methods, which means selection does not give every unit a known chance of inclusion. Of this method is 4 types. First one is judgmental, purposive sampling. Expert selects samples based on knowledge. Example: choosing sampling points near a suspected industrial contamination zone. Second one is convenience sampling. Samples selected based on ease of access. Example: testing water from easily accessible taps in a campus instead of a full survey. And then, third type is quota sampling, sampling until a specific number of each category is reached. Example: choosing 5 samples each from bottled, tap, and well water for comparing TDS. Fourth one is snowball sampling. Existing subjects help recruit more samples, mostly in social research. Example: rare in chemistry, asking small-scale gold miners to refer others for mercury exposure testing. Next, here we will see how the large sample can be reduced to small size, because small portion is enough for accurate to measure the chemical composition of substance in given unknown samples. There are 4 types of reduction methods. First one is coning and quartering, second one is rolling and quartering, and third one is riffle splitting, and fourth one rotary sample division. First, here we will learn coning and quartering. This is a classical manual method where a cone of the mixed sample is flattened, divided into 4 quarters, and 2 opposite quarters are retained to reduce sample size. Procedure: mix the sample thoroughly. Pour to form a cone. Flatten into a circular heap. Divide into 4 equal quarters. Discard 2 opposite quarters. Combine the remaining 2 quarters. Repeat until the desired sample size is reached. Examples: used for soil, grains, ore, sand, food powders. Second one is rolling and quartering. Here, the sample is spread into a strip, rolled into a cylinder, flattened, divided into 4 parts, and 2 opposite quarters are kept. Best for fine powders. Procedure: spread sample into a long strip. Roll into a cylindrical shape. Flatten the cylinder. Divide into 4 equal parts. Discard 2 opposite quarters and keep the rest. Examples: fine or sticky powder samples. Third, tie riffle splitting. Here the sample is poured through the splitter and automatically divided into equal halves. One half is used, the other is discarded. Procedure: a mechanical device with alternate slots. Sample is poured into the hopper. Slots divide it into equal halves. One half is taken as the reduced sample. Examples: used for soil, aggregates, grains. Fourth one is rotary sample division. Here the sample is uniformly splits bulk materials by distributing them into rotating radial compartments, ensuring representative subsamples, reducing segregation errors, and improving accuracy for granular or powdered samples during analytical preparation. Procedure: feed bulk sample onto divider. Start continuous rotary motion. Allow material to distribute evenly. Collect fractions in designated compartments. Combine required portions for subsample. Examples: used for large soil, grains, metals, and ore sample. Here we will learn techniques of ambient air sampling, where first one is high volume air sampling, which collects large volumes of ambient air on a glass fiber filter. Used for PM10, PM2.5, where PM is particulate matter. Example: urban air quality monitoring stations. Second one is low volume sampling. Draws smaller air volumes using a small pump and filter. Suitable for remote or rural areas with low particulate concentration. Third type is passive, diffusive air sampling. No pump, pollutants diffuse onto absorbent surfaces. Used for monitoring of NO2, SO2, O3, ozone, volatile organic compounds. Example: diffusion tubes placed at roadside or industrial sites. And fourth one is active gas sampling, impinges, absorption traps. Air is pulled through absorbing liquids, H2O2, NaOHKI, etc. Used for SO2, NH3, H2S, Cl2, O3 in ambient air. Example: Western or Indian pollution control labs. Fifth one is adsorbent tube sampling. Pollutants adsorb onto charcoal or silica tubes. Ideal for benzene, toluene, volatile organic compounds in ambient air. Sixth one is cascade impactor sampling. Separates particulates by size across multiple stages. Used for PM size distribution, coarse, fine, ultrafine. Here we will understand the technique of water sampling, where first one is grab, spot, sampling. A single sample is collected at one location and one time. Used for pH, temperature, chlorine, metals, nutrients. Example: collecting water from a tap or river at 10 am. And second one is composite sampling. Several small samples are collected over time or at different locations and mixed. Provides an average composition of water. Used for wastewater, industrial effluent monitoring. Third type of water sampling is depth, vertical sampling. Collects water at specific depths using Van Dorn camera. Example: used in lakes, reservoirs, rivers, oceans to study stratification, surface, mid, bottom layers. And fourth one is flow proportional sampling. Sample volume depends on water flow rate. Example: used in sewage treatment plants for load monitoring. Finally, here we will learn about the techniques for soil sampling, where first one is grab, spot, soil sampling. Single sample collected from a specific point. Example: used for quick analysis, contamination checks, pH, NPK analysis. Second one is composite soil sampling. Several subsamples, increments, collected from different points and mixed. Represents average soil condition of a field. Common in agriculture for fertilizer recommendation. Third one is random soil sampling. Sampling locations are chosen randomly throughout the area. Reduces bias and provides statistically representative information. And then fourth one is systematic grid soil sampling. Area divided into grids. Example: 10 meters into 10 meters, sample collected from each grid. Used for soil mapping, heavy metal distribution, GIS studies. Fifth one is stratified sampling, field divided based on characteristics, soil type, slope, land use, and samples taken from each stratum. Improves accuracy. Sixth one is incremental sampling method, ISM, many small increments collected from a large area and combined. Gives highly representative results for heterogeneous soils, example, metal contaminated sites. These are reference books for preparation of this video lecture. Lecture outlines essential steps of chemical analysis, including sampling, sample reduction, and instrumental measurement. It is highly useful for students doing dissertation or research work, as well as chemists working in industries and testing laboratories. The content strengthens understanding of accurate sampling, preparation, and analysis of environmental, food, and clinical samples. Next, please see Part 2 on sample pretreatment process, where you can learn how the collected samples can be treated to get the measurable freeforms, such as metal ions, anions, pesticide, biomolecules, and volatile organic compounds, and Part 3 on measurement of chemical substances in analytical instruments such volumetric titration, gravimetric analysis, OV, visible spectrophotometry, flame photometry, atomic absorption spectrometry, HPLC, GC, etc. Thank you for watching the Analytical Chemistry Vision. If you like this video, please visit our channel for more interesting theoretical and experimental lectures. Don't forget to like, share, and subscribe, and share your feedback in the comments to help us improve further. Thank you.